So once you sequence a genome, what does it mean? And the ability to sequence a genome has led us to a new science that we call genomics. So the genome, right, is all the DNA that's within a cell. Well, that tells us some stuff, but we also care about whether those genes are being expressed or not. Remember our gene expression chapter. So that also leads us to a concept of the proteome, all of the proteins within a cell. So we have this concept of omics, which means studying. So genomics is studying the genome, proteomics, the proteome, all of the proteins that are inside of a cell. Why would we want to know that? How could this be useful? So if we look at something like cancer, we might want to know the cancer genome, right? Remember we said cancer involves mutations that affect genes. So here's a whole bunch of different genomes of cancer. So here's a bunch of genes, and this here is looking at a bunch of different tumors. How frequently do we see errors in these genes? Like P53, we see 46% of the tumors that are sequenced here have different mutations in there. We look at other things, sometimes they're very frequent, like EGFR or KRAS here. But these different things, right, there are some that are very rare tumor types. This sequencing of the cancer genome allows us to get to personalized medicine. We could figure out ways to treat an individual's cancer that are there. But, as we said, every cell in the body, in theory, has the same genome. But not all cells are the same. So we talked about muscle cells are different from bone cells than epithelial or immune cells or blood cells. They're all different. Why are they different? Well, that has to do with the proteins that are being expressed in those cells. So there's a related field called transcriptomics. Go ahead and think about what a transcript is. What's the process of transcription? Transcription is making RNA from the DNA. So looking at all the RNAs that are in a cell, but then just because an RNA gets made doesn't mean it makes it to being a protein. So proteomics looks at all of the proteins in a sample. Many proteins are also modified, so this allows us to tell, is the protein being modified after its production? To understand proteomics, to sequence all the proteins that are in a sample, we need a specialized technology called mass spectrometry, which relies on chemistry to identify different fingerprints of certain proteins that are in there. How could we use this? Well, we could look at cancer cells, and we could look at certain proteins that are involved in cancers. Finding common proteins that are expressed or not expressed in cancer cells could find targets for drugs or vaccines that could be there. So finding different things that are activated or repressed in tumor cells allows us to identify potential drugs that could target these things. So here you can see a bunch of different drugs that target specific proteins that are overexpressed in cancer. So this idea of proteomics is new and difficult, but becoming more and more important. And not just for cancer research, but all scientific research, understanding how different cells develop and how different organisms develop and why there are differences between us and all of these things. So again, we didn't talk about this, but what do you think transcriptomics looks at? Go and pause the video, think about this. So transcriptomics, what was transcription again? DNA to RNA. Transcriptomics looks at RNA, and this is a much easier field to deal with because sequencing the RNA is much easier than proteins. But the genome is the holy grail. Sequencing the genome is great, but understanding what proteins are being expressed is the real goal, right? Figuring out which parts of the genome are expressed. Many different cells express different genes, but not all RNAs become proteins. So even though we look at transcriptomics, it might not tell us everything. Many proteins are also modified. So using proteomics with mass spectrometry, we can find these proteins and figure out how they've been modified. Using a combination of genomics and proteomics can help us understand disease like cancer, but also healthy cells and how normal development occurs. So this is just a light touch on our using these technologies. I hope you go on into future higher level biology courses and take the knowledge you've gained from this course and can apply it to these fascinating new fields. If you want to do that here at CEI, I recommend you take my Micro 250 class. We talk way more about transcriptomics, proteomics, next generation sequencing, all that stuff. But if not, I want to thank you for a great semester. And please remember, if you ever need anything now, 2 years from now, whenever, just send me an email and I will help you out. Thanks for a great semester, and it's been a pleasure. Bye for now.